Okay, thanks very much. So, what I'd like to discuss is lessons that we got from mouse models to understand human disease. And I already would like to apologize because you will not see any human patients in my studies, but you see lots of mouse patients. And in one case, you will see what happens when a mouse goes to a mouse clinic. So I will discuss three issues. First, I will discuss of the mesenchyme being a dominant mechanism in driving chronic inflammation and tissue destruction. I will uncover for you some common mechanisms that may underlie RA, IBD, joint gut axis diseases, and comorbidities. And I will discuss at the end how to properly align animal models and human disease to be able to validate hypotheses from human to mouse to human. First, I'd like to convince you that the mesenchyme is indeed a dominant mechanism in driving chronic inflammation, including rheumatoid arthritis and all the tissue destructive properties that we see in this kind of diseases. Now, I'm sure all of you will have in their mind that to explain human rheumatoid arthritis, there exists a very strong hypothesis. This is the immunological hypothesis. And in this hypothesis, we think that when we smoke, when bacteria gets into us or other allergens or challenges, then mainly our lungs are reacting to these challenges, and we have the production of these antibodies that we call ACPAs, anti-citrullinated protein antibodies, that will then bring in the immunological reaction and will lead to a more systemic immunological activity that we start destroying cartilage and bone and resulting also in synovitis. You see the linearity of this concept, and already I'd like to tell you that linear models in biology are there to fail because pathogenesis of disease is a complex process, and we have to be able to understand the many different levels and many different ways that the disease process is coming to a pathogenesis. You see at the right side here that these ACPAs, immune complexes, activate fibroblasts, and here we have a mesenchymal cell, and neutrophils that we heard very well from the last speaker lead also to fibroblast activation. Okay. This immunological hypothesis has been reviewed by several reviews in the literature, but what I'm here for today is to convince you that there is another equally or strong hypothesis that brings the mesenchymal cell into the center of the reactivities, the biological phenomena that drive chronic pathogenesis in rheumatoid arthritis and other chronic inflammatory diseases and comorbidities. So this hypothesis says that microbiota or stress and cytokines are somehow sensed by these cells, the mesenchymal cells, the fibroblasts, like the synovial fibroblasts, and synovial fibroblasts or intestinal myofibroblasts, stroma fibroblasts, mesenchymal stroma cells, even mesenchymal cells of the immune response, like follicular dendritic cells in germinal centers. These cells are the first cells to receive microbiota stress or cytokine signals, and this activity leads to effector phenomena that bring in distress in cell and tissue homeostasis, inflammation, and lymphoid tissue formation, either in secondary lymphoid organs or in ectopic lymphoid structures that form in chronic inflammatory situations. And all this we can show in many cases that are mediated by real molecules, like I show you here. Cytokines like TNF, I will be discussing today, is most of the work that we have done with this molecule, but also IL-1 and TGFs. Microbiota can be sensed by TLRs and MyD88. This is work ongoing. And intracellular signals like NF-kappa B and MAP kinase are those that mediate most of the reactivity of these cells to deliver IL-6, COX-2, hepatocyte growth factors for cell and tissue homeostasis, IL-6, MMP, cytokines for inflammation, CXCL13, VCAM, ECAM for lymphoid tissue formation. So the scenery is there. The cells are there, the challenges are there, the molecules are there, and what we need is perhaps chronic signals in these cells, especially the fibroblasts, the synovial fibroblasts in the case of RA, epigenetic imprinting on these cells that leads to persistence of this activated phenotype on the cells. It doesn't allow them to resolute, to go back in their programming and become again normal cells. Therefore, we can explain this way, in the absence of resolution, we can explain the chronicity of the disease. But you can see here that the immune response comes second, after, in our hypothesis, comes second, after the activation of the mesenchymal cell. Then we have phenomena that lead to chronic inflammation, immunity, and in some cases also cancer. This is a summary of the work that we are doing in the lab, and all we are happy to do is to see experiments validating this hypothesis. So I'll show you a few of them. Perhaps many of you will be aware that there are these TNF overexpressing transgenic mice that develop rheumatoid arthritis. These are mice that we developed early in 1991. We overexpress a transgenic TNF, human TNF in this case, in mouse tissues. And we got a rheumatoid-like polyarthritis with synovial hyperplasia, cartilage destruction, and bone erosions, mimicking much the human disease. This experiment was actually the preclinical proof of principle that TNF is causal in RA, and that anti-TNF therapies that treated these mice and make them normal also could be used in the clinic. And we published this in 1991, and then the study by Elliott et al. Came in 1993, the first successful clinical trial with anti-TNF antibodies that opened this era of biological therapies against rheumatoid arthritis, and then we have all other biological therapies coming up. In 1999, we developed another mouse model, the murine TNF overexpressing mouse. We call it TNF delta ARE. And in this mutant, you have overexpression of mouse TNF. Perhaps you have the function of both the TNF receptors, TNF receptor 1 and TNF receptor 2. And in this case, you get not only arthritis, but you get the inflammatory bowel disease, resembling very much Crohn's-like inflammatory disease. Is in fact a unique model for Crohn's inflammatory disease that is different from all other models that show the colitis phenotype. Here you have Crohn's-like symptoms, including transmural inflammation and granuloma formation, and many characteristics of the Crohn's-like inflammatory bowel disease. I'll show you that there are other models other diseases, other type of diseases may be similar in their pathogenesis because they're all driven by TNF. And we have this other mouse, for instance, the TGA86, which expresses transmembrane TNF, murine transmembrane TNF. And this gives to the mouse a phenotype of spondyloarthritis. And you can see that because you have both axial and peripheral pathology. And mainly you have this characteristic which you do not find in arthritis, which is the new bone formation, and you have it in this model. Now, a very important experiment we did back in 2001 is we took one of these mouse models and we eliminated the immune response, the immune system. So we created TNF delta ARE in this case mice, but all the mice are giving the same phenotype. Mice that overexpress TNF and get the rheumatoid arthritis phenotype. And we crossed them to RAG1 knockouts, mice that do not have B and T cells. And we could see, as you see at the bottom right, that the TNF delta A RAG1 knockout animals, despite not having any immunological response, they had full-blown arthritis. So everything was happening in the absence of the immune response in these animals. And then we got curious. If it's not the triggering of the immune response by TNF, then what it is? What is it? And we took tools from genetic engineering. These are mice, again, that with different genetic engineering approaches, you engineer them to express or not express specific molecules, like in the case I will show you TNF receptors that understand the TNF signals in specific tissues, in specific cell types. And one tool that we developed is this collagen 6 Cree mouse, which gave us a specificity for the synovial layer in the joint. You see here, very specific staining of this layer in the mouse, and also specificities for other mesenchymal cells, like the subepithelial myofibroblasts in the gut, in two different planes. Also fat cells, some keratinocytes and skin fibroblasts, and some parenchymal cells in the brain. But anyway, what we cared for in this case is that they were giving us a specificity for the synovial joint, for the synovial cell layer in the joint. So we thought, let's use this Cree. It's a recombinase that deletes sites in the chromatin that we have put LOXP sites. And we put LOXP sites around the TNF receptor. And if we are able to delete the TNF receptor to the right, delete it only from this synovial fibroblast, so we make a synovial fibroblast specific P55 TNF receptor deficient mouse, then we get no disease, indicating that the receptor is required on the synovial fibroblast to understand the TNF signal, and this is required, but also is sufficient in another experiment where we have the receptor always on the synovial fibroblast, where you get full-blown disease, is both necessary and sufficient to give you the full-blown pathology. That means that this cell is central in orchestrating all the events that we see characterizing rheumatoid arthritis, like, synovial hyperplasia, pannus formation, inflammatory infiltrations, cartilage destruction, bone erosion. In fact, I show you that the specificity of this Cree recombinase was also for intestinal mesenchymal cells, the intestinal myofibroblasts that underlie the epithelium. And in this case, again, we proved that the TNF receptor on these mesenchymal cells is both necessary and sufficient to drive the complex pathology that underlies Crohn's disease. This was a first indication that the synovial fibroblast and the intestinal myofibroblast are underlying this axis which we usually see in the clinic, which is the gut joint axis, which is the major axis in this type of diseases. Now, to convince you that this is not only a property of the TNF transgenics, because I talked only about TNF transgenics, we used another model, the Kaya model. This is collagen antibody-induced arthritis. And in this case, you see that you get no disease in comparison to animals that have the TNF receptor in their synovial fibroblasts. If the receptor is missing from the synovial fibroblasts, you get no disease. So in other models, acute in this case, you get, again, the sensitivity of the TNF reactivity on the synovial fibroblast, and not in other cell types, like we would think of in other hypotheses, like macrophages, which are responding or neutrophils that are responding very aggressively to TNF. Now, okay, synovial fibroblasts, I think I brought this cell into focus, and if you look at it closely, it resembles innate cells. It resembles macrophages, although it's not at all a macrophage, but it can understand TLRs, ligands, degradation products, cytokines, prostaglandins, growth factors, chemokines, hypoxia. Can hit this cell, and this cell will respond by enhancing adhesion degradative enzymes, matrix metalloproteases, which are so important for the pathogenesis of arthritis, lipids, growth factors, chemokines, and all these effector molecules that can actually by themselves explain the phenomena that we see in the disease, like bone erosion, cartilage destruction, immune cell recruitment and modulation, and structural homeostasis of the synovial lining. Okay, I showed you that perhaps there are interesting mechanisms that we may consider, especially when we are considering precision medicine approaches for the future. We like to have this heterogeneity because some of the patients will be of this type of pathogenesis and some of the patients will be of the other type of pathogenesis. So here you may easily see that those patients perhaps that their disease is TNF driven, at least the majority or a lot of them will follow perhaps mechanistically the pathogenesis of as it is modeled in mice. But let's see, except joint gut axis diseases, there are other comorbidities that we see in these diseases. One very evident one, which is now without any mechanism that we are discussing here, is depression, for instance, but you never know. Maybe there are common mechanisms underlying also these diseases, but mainly you can see cardiovascular diseases, as I'm sure you know, gastrointestinal, which I discussed, and pulmonary diseases. So we thought, let's take one of our models in a clinic, and we use this German mouse clinic, which is set up by this consortium, which we are members of, this InfraFrontier, which serves for phenotyping mouse patients. So you give them your mice, and they do all the tests, diagnostics, and everything, and they came up and tell us, look, your mice have a few more things except arthritis and inflammatory bowel disease. They have bone loss in the maxilla. They have salivary gland inflammation. Excuse me, they have kidney inflammation. They have interstitial lung disease. And they also have aortic valve thickening, cardiovascular disease. That was interesting. We haven't studied all of them yet, but we studied a little bit the aortic valve disease. It was interesting for the cardiovascular point of view, and you can see here that these animals develop the aortic valve, the cells in the aortic valve, which are called VICs, valve interstitial cells, proliferate, like in the synovium, and they form massive structures. And this leads also to functional dysfunctions in cardiac, as in the example here for cardiac dysfunction of cardiac output. What are these cells? And is anything else happening? Yes, you can see that you have this aortic valve fibrosis if you stain the aortic valve with Mason's trichrome. And you have evident thickening here, What are these cells? These cells are staining green by using this collagen 6 Cree mouse. So they are in the collection of cells that I described to you earlier, mesenchymal cells, and it's known that VICs, valve interstitial cells, are mesenchymal cells, but we were very interested to see that these cells are those cells that are targeted by the collagen 6, and in this case then, we could delete TNF receptor from these cells, including the synovial fibroblasts and all the other cell types in this experiment, and we could see that the aortic valve stenosis would turn, by deleting TNF receptor 1 in mesenchymal component, would turn back into normal. In fact, we could see that you could also treat these mice with Enbrel, and you could get back the normal valve histology, which indicates that indeed there are mechanisms, common cellular mechanisms, that may underlie not only the RA versus IBD, but additional comorbidities, like in this case the aortic valve thickening, which we can see in our animals, and perhaps also one can see these in human patients. In this paper, heart involvement in rheumatoid arthritis, a meta-analysis showed in yellow highlight that the strength and the grade of both pericardial and cardiac valvular involvement in RA patients, and that there is a possible relationship of these findings to the increased cardiovascular mortality of RA patients. Okay, I will finish up by discussing a little bit on how to properly align animal models and human disease. There is a lot of discussion these days, not only in scientific circles, but in funding circles, that look, mice are not so good for modeling human disease. We have so many tools now and technologies, and we can go directly to human, and we can test our hypothesis only in humans. Okay, this discussion started back in 1961 when Monod and Jacob said that anything found to be true in E. coli must also be true of elephants. And this was the excitement that they felt in those days, that the molecular biology was starting and they were understanding that genes are the basis for phenotypes. And they were thinking that if we know how genes are regulated and expressed, then we know everything. And because in those days they knew everything in E. coli, most of the information came from E. coli, they thought that they can explain elephants also in humans. So that's how it started, the argument. And it ended up these days with battles in high-impact journals like PNAS, where genomic responses in mouse models poorly mimic human inflammatory diseases in 2013, and in 2015, the same data interpreted in a different bioinformatic way, as the authors say, show that genomic responses in mouse models greatly mimic human inflammatory diseases. So data interpretation really matters. We should approach this problem and discuss it with data at hand and not just with generalizations that mouse models do not mimic human inflammatory disease. In our case, we feel also that defining the cell type that you are discussing, and in our days also the single cell types that you may be able to approach by the new technologies, will give you much more power, comparative power, and then you will be at a much better position to compare mouse cellular activities with human cellular activities. And when you can do that, perhaps you will realize that, yes, then you may be able, in some of the models, to extract information and validate mechanisms that come both from the mouse work and the human work. That's what we did in the lab. We took the mouse synovial fibroblast and we profile it in many different ways. In a way, we produced an encode, like we know in the human situation, of the of the chromatin of the arthritogenic synovial fibroblast with different assays. I'm not going to show you details of that, but we have a first map of what's happening in the mouse, and we got information from the computers of how the human synovial fibroblasts look like, so we were able to compare the human to the mouse, and we're able to see that they do not resemble so much at the gene level. This is at the gene level. You see there is no much similarity. But if you go at the KEGG pathway level, this is databases that give you genes as groups associated with specific functions, then you can see that you can reach a comparative level where both upregulated, a collection of upregulated functions or downregulated functions are similar in the mouse, the mouse and the human synovial fibroblast. By using this approach and this screening approach, we were able to fish out some microRNAs that would come out equally well in the mouse and the human situation, and this would was giving us access to epigenetic mechanisms that underlie this imprinting of the synovial fibroblast, a cell that becomes autonomous and becomes aggressive but autonomous. It's like a cancer cell. It's like a transformed cell, to put it more mildly, but it's a cell that doesn't want to die, proliferates, and perhaps underlies all the pathological issues that we see in the pathogenesis of rheumatoid arthritis. We were happy to see that, as I said, in the pathway level, most of the functions are represented in both mouse and human samples, 7 out of 30, for instance, 30%. It's a big percentage. But also we were very happy to see, and in fact in this paper that we published this year in Arthritis and Rheumatology, we were able to see that not only at the level of pathways, but also at the level of epigenetic regulation, we can see similarities between the human and the mouse. This would validate the synovial fibroblast from the TNF transgenic mice as good partner to human situation, to be able to validate hypotheses that are now growing at the epigenetic field, trying to understand how this cell is regulated epigenetically and gets this non-resolving chronic status that it gets. So, what I've shown you, perhaps, and what I've tried to convince you, that mesenchymal cell activation by innate signals, cytokines, microbiota, the example of TNF in this case, is causal, at least in the mice. It causes the disease. It's first to hit, and it's very dominant in the pathogenesis because it can drive the whole pathogenesis just by hitting one cell type, the synovial fibroblast. Mesenchymal cells that are targeted by TNF, like synovial fibroblasts, intestinal myofibroblasts, the valvular interstitial cells, these cells are targeted commonly by TNF, and they may provide a common mechanistic principle to understand chronic joint gut axis disease and the comorbidities. So we're looking forward to see additional comorbidities, whether they may have an underlying similar principle, that TNF that comes from wherever it comes, TNF may come from smoking, as you know. TNF may come from mechanical stress. It may come, of course, out of immunological response, which will explain the autoimmune basis of these diseases. But in some cases, autoimmunity will be secondary. And then first, etiopathogenic target for us will be the mesenchymal component. Our TNF mice are now validated in their biology that they do mimic human RA, so they are useful systems to validate and confirm or discover new hypotheses. And most importantly, that now it's time to use this new mesenchymal biology and inform both the therapeutic innovation and also the disease management in the context of complex chronic diseases and comorbidities. I'd like to thank the sponsors for these studies and, of course, the many good colleagues that contributed to this work, including colleagues from our spin-off company, Biomed Code Elas SA, who are using these mice to evaluate drugs and other mice, humanized mice, to evaluate drugs from the industry. So thank you very much for your attention, and I'd like to wish you having a good time in the rest of the meeting. Thanks very much for the invitation.
Thank you so much, Professor Kollias. You showed why we wanted to have you as an opening of our meeting, because you show in a very elegant way that how a career dedicated to solving a big problem and not avoiding difficult steps, like comparing mouse and human, leads to this amazing results. I had a hard time when you started, because being a T-cell immunologist, I found it really hard to understand that it's probably not that important.
I left some space. I left a lot of space for T-cell immunology.
No, thank you. So I'd like to thank you again, and you inspired me and I guess many of the people in the audience. So thank you.